Today I want to talk about the use of sensory cortex, and specifically I'll talk about visual cortex for both external and internal processing. And so to unpack that a little bit, I wasn't sure, how cognitive science-y you all will be here at a biotech campus. So I thought, let's do a quick 101 on cognitive science. And I want to talk mostly about these three terms. So perception, attention, and working memory. So if we start with perception, it's pretty straightforward. And as you will notice, you'll have intuitions about all of these. So there's some visual input in this case, and you're processing that input. If you have to pay attention, for example, if you see the stimulus on the screen and you have to make, let's say, an orientation discrimination, you may pay attention to that stimulus, which you can, or I've highlighted here as a sort of a spotlight. This is a pretty common analogy. Now, if we take that stimulus away from the screen and we ask you to now remember it, we would usually call this working memory. But as I'm talking, the stimulus has been gone for a long time and a longer time and a longer time. So you could also say, well, at some point, this crosses the border from working memory or short-term memory into this longer-term-ish memory. Now, to make matters maybe worse, you can also say, okay, now I want you to manipulate this thing in your mind, which you could call working memory, but you could also call it visual imagery, because now you have to imagine something that wasn't there before and that wasn't part of your input. You can also say, okay, this is the thing you should expect to see on the screen later. So now we call this expectation. The point I'm trying to make is that in cognitive science, one of these tendencies that seems to be happening is that people have their little topic and they study that and they call it either working memory, long-term memory, imagery, short-term memory, expectation. And I kind of want to break away from that. So to me, I think these borders are very blurry, like when is long, when is short. If you manipulate it, is that working memory? Is that now imagery? So I'm going to agree, hopefully we can all agree on this, that they can categorize these things into roughly two classes. One, things that are out there, yet are coming in from the eyes. And two, things that are in here, so that are happening in the brain, that are happening there without input. So I'm going to lump these all together for the rest of the talk a little bit. Now, the next thing I want to briefly introduce is the concept of sensory recruitment. But before I do that, I want to point out something pretty obvious, which is that we have this part of the brain, the back of our brain, called visual cortex. And this, what I highlighted here is primary visual cortex, area V1. And this part of the brain is very important for perception. In fact, it's the first cortical input or the first cortical site where visual input is processed. Now, the idea of sensory recruitment is that this part of the brain actually also reflects information when you're attending something. So maybe you're attending one visual stimulus and not another. You see that that is reflected in patterns of activity or even just wholesale activity in early visual cortex. Similarly, and this was work from 2009, people have shown that even when there's nothing on the screen, if you ask people to remember something, then in these patterns of activity in this primary visual cortex, you can see and you can predict what the stimulus is that someone is holding in mind. And that's just a few examples. As I already showed on the previous slide, there's also retrieving things from long-term memory, there's visual mental imagery, there's expectations. All of these concepts have kind of been shown to draw upon this part of the brain. So we can basically establish that V1, primary visual cortex, does everything, and we don't need the rest of the brain anymore. Yeah? That's a little bit, of course, a joke. I'm not serious, but it does beg the question that how can this be, right? Because V1 is for seeing. That's how we always conceptualize it. Why is it now that in cognitive science, everyone is saying that V1 is used for cognitive functions, for maintaining or somehow holding in mind visual information? Oh yeah, that's my joke. Sorry, it's got on the slide a little bit too late. All right, so for the rest of my talk, these are kind of the overarching questions that I want to address. So why, but also how are these primary visual cortices representing these internal mental contents? And sometimes I may slip because people say I'm a working memory expert, but I am trying to veer a little bit away from that and talk more about these internal mental content. Can be many things, working memory, as we classically know it, just being one of them. All right. So I want to give you just a little bit of background of how I arrived at this main question, and also just show you a little bit of what V1 can do and also what it cannot do. So what I've shown in the past is that if someone is holding in mind a visual stimulus, such as the orientation of this grading, if we try to decode that visual mental content, we can do that. And we can do that if there's no sensory input on the screen, but also if we show some kind of noisy stimulation on the screen, and even if we show a second grading that's irrelevant to the task, also on the screen. So what we've shown there in the past is that we have this distractor-resistant visual memory representations, so that this part of the cortex can even hold these things that you have in mind when there is visual input incoming. Now, one other cool little detail of this paper is that when we try to decode the actual visual input, of course, we can do this as well, right? V1 is still a primary sensory region. So that means that we can simultaneously represent both the content of perception and of memory. But what happens when we add other cognitive factors like attention? So we show this stimulus, but now on every single trial, when subjects are remembering an orientation, they may also be asked to attend the orientation that's on the screen or attend its contrast, actually. So quickly, we try to decode now the orientation that people are holding in mind. Can we do that? I'm not sure if you can see my mouse. Yeah. So when you don't have to attend the information on the screen, yes, we can. But when we ask people now to attend the stimulus that's being shown to them, either look at the contrast changes or the orientation changes, then we see that there's a huge drop in the decoding of this remembered orientation. Vice versa, if we now try to decode the stimulus that is on the screen, what we see is that now with attention, we actually see an increase. So we see a boost in decoding of the orientation that's on the screen. So I want to make a little bit of a leap here. If it's true that working memory and attention cannot happen in this part of the brain at the same time, so there's this trade-off between these competing top-down demands, then it's possible that a bunch of cognitive functions, maybe all of them, that they overlap or at least draw on the same resources in this part of cortex. So these visual mental contents, we know that they exist in V1, but maybe you can only hold one memory or attention or anything else at the same time. So there's limits there. Okay, so now I want to delve into some recent experimental work from my lab. And the first question I want to get at is why to recruit V1 at all for these visual mental content? Yeah, so there's like why and how, but let's start with why. And let's see if we can answer this. So based on some previous work, we had the hypothesis that, well, maybe what V1 is doing, maybe it's acting as a sort of comparison circuit. Because, of course, visual input is coming in a visual format. So if you have this visual mental content in a visual format as well, then V1 might actually be a very efficient site if you want to make high precision comparisons at the earliest possible opportunity. So this might be just the perfect place to do that, perfect place in the brain. And I want to highlight that this is work largely done by R.L.Rademaker in my lab, who did all the scans and all the analysis. And this is actually what she did in terms of the task. So subjects came in, they did 4 sessions in the scanner. We have 15 subjects. And this was basically their task. We showed them 2 templates. Now what subjects had to do, there's these moving dots, but they're moving in a given direction. And they get these 2 dot motions, 2 different directions. And then we post cue them to remember one or the other direction. So this is a direction of motion memory task, or visual mental content task, if you want. So they get this mental template. In this case, it's these dots moving into the upper right direction, and they have to hold that direction in mind for 7 minutes. Yeah. Now, we don't just keep them there bored for 7 minutes, we also give them a little extra task. And that is that after some variable delay, they get a little cue. In this case, that cue is green. And that means that, watch out, another direction of motion is coming, and you're going to have to compare it to the one that you're holding in mind. You have to say whether it's clockwise or counterclockwise. And so throughout these 7 minutes, there's 24 of these comparisons. Now, we also have another cue color, in this case, red. And that means to the subject that now you don't have to compare anything, but we want to control for motor output. So we just tell them, like, if you see this red fixation dot, then you just press a third button. So not clockwise or counterclockwise, but just another button. They also do this 24 times throughout these 7 minutes, and we randomly interleave the order in which these comparisons or non-comparisons are happening. Now, importantly for later analysis is that this this template and probe directions are independent. So with template, I mean the thing that they're holding in mind. With probe, I mean all these comparisons that they're making. And at the very, very end of the trial, sorry, that hurts. They have to recall the actual direction of motion that they saw at the very, very beginning. Yeah? Okay. Then, because this is such a long delay of 7 minutes, now you start to understand also why subjects had to be in the scanner for different times. So what we do is we do this across 32 blocks, and we show them 32 unique templates across all these days of scanning. All right. So let's start with this main task. And what does it test? What are we looking at here? So to go back a little bit to my introduction is that, well, we are looking at perception because subjects are seeing all these different probes and they have to look at, which direction of motion they're going. But we're also looking at attention. I already kind of said it. They don't only passively perceive these thoughts, they have to figure out the direction of motion. They have to do working memory, if you will, because they have to hold this template in mind throughout. But they do that for 7 minutes. And no one's really done that before. Like we usually go up to 12 seconds, maybe 15 if we're feeling adventurous, right? But 7 minutes is pretty extreme. And more. I mean, they're also doing sort of comparisons. They are maybe expecting to do something. They may try this comparison versus not comparison. Who knows what they're doing then? So point being, again, that when you think about it in terms of function, then all these concepts become very blurry. In this case, what subjects have to do is make comparison and say if it's clockwise or counterclockwise, or give a button press. And what kind of task that is, I mean, maybe you can tell me afterwards. Yeah? All right. So then what did we predict? So first of all, when we have these sensory inputs that are happening all the time, we call this the probe, we would predict that this would be decodable from B1. So its direction of motion should be decodable from B1, whether or not it's used as a comparison. Why? Because we're looking at B1, primary site for visual input. This is visual input. It should care about that. With respect to the mental template, and this is where our comparison hypothesis kind of comes about, we would expect that you can only decode the direction of motion when you're actually using that for your behavioral task, when you're making a comparison. And we think that we should probably not be able to decode this, although we don't know, right? We've never gone up to 7 minutes, but if you're not doing anything, then maybe we'll also not be able to decode that. Now, a little thing that I didn't mention before yet, when these comparison stimuli come on the screen, we're actually also manipulating the motion coherence. So there are some trials where those dots, they actually have no motion coherence. We don't tell the subjects this, we just say there's some trials that are really, really hard. And they did this for days, right? Most of them didn't really catch on to that. They tried to really do the task. Sometimes the dots are 50% coherent, and sometimes they're all moving in the same direction, 100% coherence. So we have some predictions here as well. There's a reason we did this. All right, so in terms of sensory input, if there is no coherence, we wouldn't predict we can decode the direction of motion. I mean, we only have a label in our experimental program, but we don't actually have a direction of motion on the screen. And then we predict that as the coherence goes up, we should also be able to better decode the direction of motion. However, when we look at the mental template, the thing that people are holding in mind, we would predict that because you're trying to do this task, you're trying to make this comparison, we would expect that you can decode the direction of motion that someone is holding in mind, irrespective of what the coherence is of the visual input. So far, so good? All right. So did participants do the task? I think that's a fair question because we were a little bit worried about that too. We thought, okay, we tell them, don't compare, don't think of a pink elephant. But first of all, the template. Did they recall it pretty well after 7 minutes, which is a long, long time? They did. Yeah, so what I'm plotting here is just an error distribution. So the frequency of a certain error occurring, and you see that the peak is at 0. So most of the time, people's responses were centered on the thing they were holding in mind. Now, did they think of the pink elephant? Looks like probably after a while, they got too bored to think of the pink elephant. So we see that, first of all, they press the right button most of the time, like 90% of the time. When they do make a comparison, they're quite accurate. So their accuracy is at 9.4%, so they were engaged with the task. And the most interesting here to look at for these comparison versus non-comparison trials is the reaction time. So if they were somehow, not trying to do the task, they had figured out that this direction of motion was completely random when it was 0% coherent, then they would just quickly move on probably. And if they are not doing a comparison, or if they're still doing comparisons, then you also don't expect a difference between trials where they're making comparison and not. Let me unpack that. So when subjects were told not to make a comparison, their reaction times were really, really fast. And they were equally fast for all motion coherences. When they were asked to make a comparison, their reaction time was much larger. So that tells us that they were probably doing something in one case and not in the other. Moreover, and this is not significant, but if anything, in the 0% coherence, they took longer. So they seem to have been trying to at least figure it out. It wasn't that they were like, okay, well, this thing is random. I'm just going to move on. I'm not going to try my best. Okay. Now we get to the neural data. So what did we do for our decoder? We actually had a separate set of runs where we did a separate task. We call this our sensory mapping task. So we show, again, direction of motion stimuli with shuffled ITIs. We did that for 256 trials, and we actually asked them to do an orthogonal task, so they had to do some contrast detection tasks. So we wanted to train our model on these data. And the second point here was to use this to localize visually responsive voxels, because if we're just going to look across entire V1, then we're also going to include a lot of voxels that are just doing noise. So these stimuli are used, 1, to localize visually responsive voxels, and 2, to train our decoder. Okay, so what does the decoding look like? So what I'm plotting here will be decoding as a function of time. And I will show this both for the sensory input on the left and for the mental template on the right. So to orient you, this is the time, so this gray square, this is the time when the comparison stimulus was on the screen. And if we look for comparison and non-comparison trials, never mind. This, because of our jittered time between all these comparison stimuli, this is the time where the next possible stimulus might happen, the next possible comparison stimulus. And the next one after that, because some of the ITIs are longer than others, and then the third one isn't there. But the take-home point here is that if we look in response to the subjects making a comparison, seeing one of these comparison stimuli, what we see is that we can decode very well the direction of motion of the visual input, and we can decode that roughly equally well, whether or not they're making a comparison. There might be like one time point where it's a little bit better if they are making a comparison, so when they are attending that stimulus. The mental template is where things really get interesting. So now we're trying to decode the thing that they're holding in mind. When they are making a comparison between that mental template and the item that's on the screen, we see that we can decode the mental template. However, when we're asking them, withhold, don't make a comparison, press this third button, we see that we can no longer decode the direction of motion that they're holding in mind. So the next thing we do is we estimated the hemodynamic response function shape, and we fit it to each voxel and each trial to estimate then the bold amplitude in response to each of these comparison stimuli. And what that gives us is these bar plots. And we see basically the same thing, just collapse now over time. So when we are decoding the sensory input is represented independently of task and also irrespective of task. But the mental content, the thing that they're holding in mind, is only represented when it's used for a comparison. Now, I also told you about these different levels of motion coherence. So what about those? So let's again start with the sensory input, where we had a clear prediction, right? We would expect to see higher decoding if we have higher motion coherence. And when people are making a comparison, we see that that's indeed the case. Yeah, so there is no decoding, obviously, when there's no motion coherence. And this is higher when the motion coherence is higher. Arguably, we could have gone a little bit lower with our middle condition because it's already kind of pretty good there. And when subjects are not making a comparison, so arguably maybe not attending the stimulus as much, we see the same thing. So that there's this graded increase in decoding with increase in dot coherence. Now, the mental template, which is, of course, the most interesting part here. When they're making a comparison, we saw that we can decode this mental content. Is this dependent on the coherence of the visual input or not? And the answer is not. So we can decode the template, the thing that they're holding in mind, equally well, and it doesn't matter what the input is. So even when the input is completely random, there is no direction of motion at all, we can decode what people are holding in mind, and they're trying to do this comparison. And of course, as we already saw before, when they're not making a comparison, there is no decoding of the mental template. So sensory input depends, the coding of sensory input depends on this probe coherence, whereas the coding of the mental template depends on whether or not they're making a comparison, but not at all related to the coherence of the probe. All right, so as I was sort of hypothesizing at the beginning of this, is V1 actually a sort of comparison circuit? That might be going a little bit too far based on these data, but I do think that we have fairly strong evidence to suggest that these mental representations, they recruit V1 when they are compared against input, but otherwise not. I also want to argue a kind of broader point is that if we want to understand how information is represented in the brain, irrespective of the cognitive task that, or however we want to name it, we really need to consider how that information is used, because we're asking the subject to do something, and that is probably driving the way that they are harnessing their brain power and which parts of the brain they might recruit to do that task. And so a very closely related point is that when we focus on the function, the thing we ask, the thing that the brain is built for, then these blurry lines between all these concepts will become much, much less relevant. Now, we talked about kind of why. So why might V1 be doing this? And tentatively, I think my answer would be as well, when you need it, you will recruit it. Yeah, so you will use it to compare whatever you're holding in mind with something that's out in the real world. Now, going back again to somewhat shorter delays, we know that we can decode these working memory contents. We know that this is a visual part of the brain. We can decode visual contents from there as well. So what is that? It's not possible that these things are exactly the same, right? If you're looking at someone's face, you have input. And if you are remembering the face of that person, it's clearly different. Right. So what are these mental contents? Are they just, and this is a little bit of a straw man, I'll admit, but are they just noisier versions of perception? And this is, what I kind of already alluded to a few times is that we're talking here about V1. It's the first cortical target of visual input. So of course it's going to do visual perception, but now it also apparently is doing a lot of this visual mental content. Why? So how is that possible? They must not be the same thing. They're experientially not the same thing. So the next question we asked is that, okay, if we sort of intuitively sense that these are not the same thing, do they maybe have a different format? Are they maybe noisier? Do they maybe have a different format? And if so, we assume that that's the case. How does that, this internal mental content, how does that format differ from when we're perceiving something? And so this is worked together with Chaipat Chunhras, who's now actually a professor at Chulalongkorn University in Bangkok. But we were together back in San Diego. And so this is actually one of our pandemic projects when we wanted to reanalyze some data. And so we have this very simple task. I already showed it at the very beginning. This is this task where we showed that we can decode working memory contents, despite if there's something on the screen that's either noisy or another grading. So here we just said, okay, we're going to take all those data, we're going to collapse across them, and we're going to use that as representations, like working memory representations or visual mental content representations. What I didn't say yet is that during that study, we also showed people these gradings, just again, as this sort of independent mapping task. And so in the sensory task, what subjects were doing, they had this 9 seconds, there was a grading on the screen, it was phase reversing, and they were attending contrast changes. Again, deliberately, this is an orthogonal task because we only want to look at sort of this passive input to the visual system. What kind of responses do we get there, and compare it with these active sort of working memory representations when you're holding something in mind. Now, the next slide, I'm not sure if that's necessary, but for those of you who've never done representational similarity analysis before, this is a little bit the idea. So if I put you in the scanner, I show you a face. This is my friend Sirwa. She makes a funny face. I show you that face again, and now I correlate the patterns of responses that I measure. I might get a really high correlation because every time you see that face, you get very similar responses. Now I show his face again, but now he's wearing an EEG cap. Still pretty similar responses, but now I'll show you some gazebos. And if I correlate that with the pattern of the face, it's actually quite dissimilar. Yeah? So I can fill out this entire matrix. So faces with faces, pretty well correlated. Faces with gazebos, not so well correlated. And gazebos with gazebos, also, again, pretty highly correlated. And so any area of the brain that would represent faces versus gazebos, you would see, or you might expect a pattern that looks a little bit like this. So that's RSA in a nutshell. Now you can build models, make it interesting. Some toy models here, completely, I made this up. This is not real. This is not real data. One model, you could say, okay, the brain is only going to respond to specific exemplars, but it's not going to generalize from one face to another. Yeah. Or you're going to say, well, but this is an objecty area. It's going to do faces versus gazebos. And then you can, of course, correlate these models with your data, and then you have an experiment. So that's what we're going to do. Except that we're going to use orientation. So it gets a little bit more complicated, but the idea is exactly the same. So we have some number of trials, and we have some number of voxels. And the first thing we do is we're going to split these in half. So we now have half the trials on one hand, half the other trials on the other hand. What we do then is, because we have 180 orientations in orientation space, we're going to bin them a little bit. So we're going to take any orientation plus minus 10 degrees, and we're going to do that in one half of the data. And then for each orientation, we're going to correlate that response vector with every other binned response vector in the other half of the data. So what that gives us is a correlation between an orientation of 1 degree and another orientation of 1 degree in the other half of the data, and the next one over, and the next one over, and so forth. Yeah. So we get a representational similarity matrix out of that. Of course, we only split the data randomly once. That would be pretty noisy. So we do that actually 1,000 times. We also do this for each subject individually, and we do this for each region of interest individually. Now, before I show you any of the data, I just want to remind you, and this is maybe very obvious, but it's good to think about what can we expect here, is that orientation, physical orientation, is continuous. So it's not categorical like faces and gazebos, it's actually continuous. So an orientation that's like this is very similar to the next one, but it might be quite dissimilar to the one after that. I have a little demonstration of that. So let's say 42 degrees, you would expect a very similar response if you show that 42 degrees again in an area that cares about orientation, of course. But also an orientation of 50 degrees might give you still a very similar response. Now, if I show an orthogonal orientation, so 125 degrees, you would expect much lower similarity. Yeah, so this should give rise to what you see here on the left, which is kind of a diagonal that then smoothly tapers off as a function of distance and orientation space. Okay, here come the data. So again, this was a pandemic project, and we show you this data now, and then there's like 2 years of analysis that happens after that to quantify it. But this was the first thing we saw, and we thought it was super cool. So let's look at the sensory task. So when there's this input, passive input coming into the visual system, and what I'm going to plot here is a bunch of different visual areas. So V1 here is now all the way on the left. So the very first one, and then we go up the visual hierarchy. We even have some object areas here and some IPS parietal regions. Yeah. Okay. So when we did this, we thought, okay, that's cool. That's kind of what we expected. There's a strong diagonal, and we see that it tapers off a little bit. Of course, we get these hot spots in the corner because it's orientation space, right? It wraps around itself. And then we thought, okay, it gets a little bit noisy up here, but okay, we know that it's sort of not attending orientation, so maybe IPS doesn't care so much, but this looks cool. So let's look at the memory task. Might it look the same? Might the format of representation be the same? And so this to us was extremely striking. This looks very different. And the most notable thing that you might see straight away is that you get these two kind of blobs. So, and they're centered on the oblique orientation, so 45 and 135. So what that kind of tells us is that anything that's not a cardinal, and it's in kind of one of these orientation quadrants, they all kind of get lumped together, becomes very similar. And then all the orientations in the other quadrant also all get lumped together. They become very similar. So this was like the quick analysis we run, and now comes the rest of the story, or at least the short part of it. How can we quantify this in a principal way? Because it's a very in your face. It's clearly a different format. We see that there's some kind of categorization happening. How can we actually quantify this in a meaningful way? And so what we decided to do was base ourselves in visual input statistics. And so if I have a scene such as this, and this happens to be the Frankfurt skyline, one thing you might notice is that there's a lot of vertical and horizontal orientations. Now, even if I don't look at the Frankfurt skyline, of course, you're saying I'm cheating, because of course you're going to see a lot of these orientations, but even in natural scenes, and other people have done this long before we thought about it, of course, if you highlight all the parts of the image here in red that have clear orientation content, and you make a frequency histogram of which orientations occur more often, what you see is that there's a lot of cardinal or horizontal and horizontal and vertical orientations. And there's just fewer of these oblique orientations. So these kind of orientations. Yeah. So we thought, okay, we'll use these visual input statistics as the basis of our model. But before I continue, I want to make a very quick demonstration to show that this is, you might think, oh, this is all a little bit esoteric, but this is very meaningful in a perceptual way. Yeah, so bear with me. I'm going to ask you all to do the following task. So if you're used to psychophysics, you're already fixating. If you're not, please fixate the red dot. So what I will do, I will take a part of this scene that you just saw. I'll just crop out one of these buildings. And the building can have one of 180 orientations. And I'll show it at a given orientation. And then I will show it again, after a delay, a second time. And I want you to clap if the orientation is the same. I want you to stomp if the orientation is different. Yeah? All right. So here comes the task. I'm not sure everyone is awake. Let's try that again. All right. Wait, did I say stomp is different? Stomp is different. Okay, good. Okay, good. Maybe I am not awake. I thought, why are they not clapping? They were different. Yeah? Okay, so now we got used to this task. Let's do another trial. Now you know what to expect. I'll again show two of these buildings, one after the other. Clap if they're the same, stomp if they're different. Yeah? Here it goes. Ah, good. I love when a visual system works for me. Good. So I did one trick. I put these buildings either close to vertical or I put them close to an oblique. But the difference between them was exactly the same both times. Yeah, it was only 5 degrees different, which is pretty small. So Actually, I shouldn't have insulted you earlier. You did really well on that first task. It was really more my fault. But the difference between them, and you're just going to have to believe me, or, oh, we can do it again. Okay, hold on. I have an idea. Tilt your head so that it's now aligned with those two buildings. All right, we try again. Did you see now that they were different? I see some, okay, we can do it one more time just for the sake of, yeah? So make sure that it's vertical for you, yeah? Then it's gonna be easier. Okay, good. I'm glad that worked. Yeah, so I don't have to tell you, just believe me, we actually just experimentally showed that if something is close to one of these cardinal orientations, your sensitivity is just much higher. So these visual input statistics are not just some kind of cool quirk, but your entire visual system has adapted to integrate those statistics, and you're just much more sensitive to those things that you see more often. Yeah, this has a real noticeable impact. This is just to justify our modeling approach a little bit. Okay, so what did we do with the modeling? So we take this input statistics function, and we say, okay, now we're going to make two models. One, we want to have something that, what would the visual system do, kind of in fMRI. So we take 180 von Mises functions, so to sort of simulate the tuning of differently tuned voxels. In this case, they're not neurons, they're voxels. We're talking about fMRI. And we're just going to tweak their amplitude based on where they are in this orientation space. So previous work has shown that there's higher amplitudes when you're showing these cardinal orientations versus the oblique orientation. So that's all we're doing. We're just, twisting one knob. And then what we can do, we have this bank of filters, this bank of von Mises functions, and we can present an orientation to it, and we can read out the response. Then we can correlate that response to every other response to every other orientation. And so what do we end up with? We end up with this, we call this our veridical model, our veridical representational similarity matrix. So this is model one. We say, okay, if the visual system does what we think it does, this is kind of what the data might look like. Now, for the working memory task, we saw that there's this strong clustering. So we thought, okay, there's some kind of categorization happening. How might that be happening? So we again take the exact same input function. And now what we do is we calculate something that we call the psychological distance. It's basically just the distance you have to travel along this curve to get from one orientation to another. So to demonstrate here, these two blue dots, those are those two upright buildings. Yeah, those are orientations that are only 5 degrees apart, but there's a lot of space between them if you have to travel along this curve. So we just take the integral along this curve between any two pair of orientations. Similarly, if you're in this oblique part of the space, then actually there's not so far you have to travel. So your perceived similarity is much higher. So the psychological distance between those two things is small. They appear the same. The psychological distance between these two things is high because they appear very dissimilar. Yeah. And so, like I said, we take the distance between any two pair of orientations, and then we end up with this categorical model over here. So now we have two models, and we fit them to first the sensory task. Yeah. So when we do that, what we see is that the veridical model here plotted in gray does much better at explaining the data than this categorical model. Now, where it gets really cool, of course, is this memory task where we saw these blobs. So now we see exactly the opposite pattern where the categorical model does a much better job explaining the data. And I think it's also really neat that it seems to be doing an increasingly better job as we go up the visual hierarchy. And so we know that this must be some kind of feedback signal, right? Otherwise, this wouldn't work. There's no input after all. So we see that this is especially strong in area before IPS 0, etc. All right. And then we did many other things. And this is just one example because you might say, okay, well, this input function you use is kind of whatever, it seems a little bit arbitrary. Could I do something like this for my experiment where I don't know the input function? And so here we use a separate psychophysics experiment, separate set of subjects. And all we did is we gave them trials with different orientations. And every trial they had to recall the orientation. So they had a little method of adjustment task, and they had to reply, like, what orientation did you just saw? We had them do many, many, many trials and many subjects. And what we generated was this function here where we just plot the absolute error of people's responses as a function of the target orientation that they saw. And so what we get is, this is just data. So this function, you see that is much higher errors. It's to the power of -1, much lower errors for these cardinal orientations. So they're much better there, but the errors there are much higher in these troughs, so around these cardinal orientations. And so instead of using this arbitrary input function, we now use one that's got from independent data in the psychophysics lab. We again generate our models. They look kind of similar, and also the results again look very similar. Okay. So here we see that if you have perceived orientations, those are largely represented veridically, at least as we define with our veridical model. Whereas these remembered orientation, they become gradually more categorical along the visual hierarchy. I hope that this is also a third point that you'll take away from this, is that these input statistics, they have really high potential to model perception and memory of many visual features. Right, because we often use orientation and direction of motion because they're quite well characterized. We kind of know what they do in visual cortex. But once you go to another feature like color or faces or objects even, if you could just map this psychophysically and you can generate a model from that, you can also then scan that and look at the differences. And if there are these kind of troughs in the landscape of the sensitivity landscape, that can then explain how your data might be categorized when you have to hold it in mind as a mental representation. Let's do a little choose your own adventure. Let's see how this kind of work out. So I have 2 more parts. One is about kind of the implementation. If we're talking about early visual cortex, how might feedback or other mechanisms contribute to the signals we see there? How might that be implemented sort of in the nitty-gritty of the brain? So I have some theoretical work there and like a short teaser on some experimental work. Then the other part is about eye movement. So I don't know about you all, but when the lockdowns and everything were happening, my lab pivoted a little bit. So we started doing some eye movement work. So there we look at, again, distraction during visual working memory, and we look now also at statistics, but statistics of eye movements. So those are the 2 things I still have. So we have sort of theoretical modeling, implementation of how things might work in visual cortex, and we have eye movements and distraction, kind of more sort of unrelated maybe, but also fun. Okay, who would like to hear about modeling theoretical implementation? Okay, yeah. Who would like to hear about eye movements? Okay, sorry, I think the theoretical people won. Yeah, but it was close. It's close. All right, good. Then we continue kind of in the same theme as we've been going. So how might mental content recruit sensory areas? And again, I want to start with a hypothesis. So we hypothesized, and that I mean myself and one of my PhD students, Noah Krause, who is on the picture here, and who actually also again implemented the work I'm gonna show you. We hypothesized that there's feedback signals from frontal areas, and this is not super new, right? I already talked about recruitment and sensory recruitment and things like that, but that there's these feedback signals from frontal areas that direct subthreshold signals in V1. And that by making them subthreshold, or by them being subthreshold, we can dissociate what is happening inside the brain, because it's sort of also, again, a different format, a different implementation from what's happening out there in the world. To go back to that very early distinction where I hope we were all on the same page, that there's stuff out there, stuff in here. Yeah. But also that this may allow flexible functional adaptation to whatever the task might be. And I might even go one step further. When we talked about comparison, this may even sort of be a way to implement some kind of filter on your incoming information. Yeah. So first, why am I talking about subthreshold signals? I don't think we talked about that up until this point. So there are some reasons to suspect that in the literature. But, I'm happy to debate that. In fMRI, and this I showed you at the very beginning, what we can do, we can decode the visual mental content well, maybe not in 7-minute paradigm, but in these like 12-second paradigms, we can decode what people are holding in mind pretty much throughout the entire delay. So what does that mean? That means that these signals that we measure here when people are perceiving a stimulus, they are overlapping well enough with the patterns that we record when someone is holding something in mind that we can cross-generalize between them, and we can do that kind of throughout a delay. So that would imply sort of a sustained signal. But if we now take another measurement technique, we use EEG, what we see there is that there's really only transient decoding. Well, first of all, in response to a visual stimulus, right? You get the transient signal and you can decode the orientation of that stimulus. And then when we take that stimulus away and you're just holding it in mind, the kind of decoding goes away. So what do I mean with in response to an impulse? I mean, if we show something like this, so we show some neutral content, and when we do, that then synchronizes the recordings again, and we can again briefly decode what was there in mind, yeah? If we look now at literature from electrophysiology, so from monkeys, macaque, what you often see is that there's no decoding from spikes. So we look at spikes, we get a lot of responses, very similar to EEG, get a lot of responses when you show a stimulus that's transient, goes away, but then during the delay, there's actually not so much happening. However, there is some work that has shown that if you look at the LFP, the local field potential, that then again, you might recover information about the visual mental content, yeah? So there might be a little bit of a dissociation here between electrical signals that are spike related, and tentatively, yeah? And the LFP, sort of what's in the soup, what might be measurable if you take all of the stuff together. All right. So if we for now say, yeah, it's pretty likely that those things are subthreshold. And actually, this is not anecdotally entirely, but we're working on a monkey data set now where we have recordings from V1 during memory task. There's not much going on during the delay. And so we're looking at this as well. So I'm quite convinced of this. So I think you might want to be as well. But, everything with caveats in science. If we assume that there are these subthreshold signals, so what we are decoding is somehow subthreshold, where is that coming from? So one hypothesis, and originally this has actually been proposed as a mechanism for frontal cortex, but one mechanism might be short-term synaptic plasticity. And this refers to a change in functional connectivity in response to previous firing. So what does that mean? What do I mean by that? Is that if you have a network of some neurons and you present, let's say, a stimulus to that, you get some kind of firing in some of these neurons. And when they fire, you also see that the connections between them become a little bit stronger. So once you take that stimulus away, you have this kind of potentiated state in the network. So that if you drive that network again with some neutral input, now these potentiated neurons, they're going to be more likely to fire. Yeah? So it's really this activity-induced change in connectivity that then when you wash some more information through it, you can kind of ping it back out, if you will. Now, the alternative, and this I've already talked about a few times, and it was also already in our hypothesis, what we think might be going on, maybe something more like feedback. So you have feedback signals from higher level cortex that are somehow impinging on early visual cortex. And this allows the memories or the visual mental content to be, for example, distractor resistant, as I showed at the very beginning, which we saw in fMRI, right? So even if you have a big thing flashing on the screen, we can still decode what you're holding in mind. And what it also allows us to do is to really make this task relevant. So you could flexibly allocate, let's say, you hold one thing in mind, but not the other. So that would be the benefits of having something like feedback. Now, I think these ideas are around in the literature, but haven't really been formalized. So what I'll show you next is really a formalization of this, a kind of a proof of principle, if you will. So how did we do this? We came up with a model that's based around simple ring attractors. So we basically simulate the firing rates in this neural network model. So we have an excitatory ring here consisting of 180 units, and those are connected through sort of a micey-shaped excitatory connections. And then, of course, there's also an inhibitory ring, which then inhibits based on how much excitation there is in the ring up here. So this is very sort of basic building block, a ring attractor that have been used in many studies. Now, what we did with this sort of basic module, we tried to make now a mini brain. And of course, this is no brain in any real sense, but we came up with like left hemisphere, right hemisphere for early visual cortex here in blue, and then left hemisphere, right hemisphere for frontal cortex. We have some mutual inhibition between the hemispheres and frontal cortex. And of course, signals can come in to the visual cortices. So one orientation maybe to the left hemisphere, another to the right hemisphere. And then we have a cueing mechanism, and we actually have several, but this is just sort of the most basic flavor of this, where we can cue then afterwards whether we want this one or that one for the behavioral output later on. Okay. Now, on top of this basic architecture, we have either STSP, so short-term synaptic potentiation. And so we do this by having sort of resources in calcium. So when you fire these neurons, you deplete the resources, but you get an increase in calcium. And that increase in calcium is what's going to give you kind of this continued content, right? This synaptic strengthening. And in a second version of this model, again, these are the most simplest versions I'm showing you, we add feedback. So here we have left and right frontal cortex sort of feeding back what they have to these early visual cortices. All right, so that's the basic architecture. Now I want to show you just an example trial. So what people have shown before is that you have these ring attractors. If you insert some information, they are able to sustain that information also when it's no longer present. And so that's what I'm showing here in the lower left corner in this frontal ring. So this frontal ring was actually tuned to have these attractor dynamics. Yeah. So we show a target. So let's say an orientation. We show that first comes through early visual cortex, then it goes to frontal cortex. And in frontal cortex, you see that this representation is maintained. What we see in early visual cortex, which we tuned to only have these transient dynamics, we see that there's a response, but that response then goes away. So we basically just take the same kind of module as we do in frontal cortex, but we make everything a little bit less. We dial it down so that we don't get these attractor dynamics. Yeah. All right. So after we do that, we show one of these impulses to see, okay, can we indeed ping out what was being held in mind? And again, this has been done before with this STSP model. People have shown this, and we see this as well. So when we present an impulse, first of all, we see that it comes into visual cortex, right? And because it's has no real orientation content, what's happening is that we see firing everywhere in all of the neurons, not depending on what orientation they're tuned to. But we do see that we see this potentiated response to the orientations that were shown, to the target orientation that was shown earlier. Yeah? Okay. Now, what hasn't been shown before, because people have said, well, you need STSP to have this kind of revival if you are putting an impulse into your network to get that information back out. STSP, that's what that's for. But no one has shown before that you can get exactly the same if you use a feedback model, and that's what we show here. So again, we have this visual input, comes into early visual cortex, gets sent through to frontal cortex, where we get now this bump attractor that's sustained. But when we now give an impulse, because there is this feedback, we still get this potentiation, or we have this potentiated state in this early visual cortex that still gives this differential response. So we can, again, ping back out the target orientation that we showed at the very beginning. Yeah? All right. So what we show here is that the early visual cortex of both of these models, they can keep information in these subthreshold states, and they respond to perturbation, which means that we can get that information back out by sending an impulse through the system. So then the next thing we did is we want to give some tasks to this model. And as I said, this is a little bit of a proof of principle. These are tasks that we've seen already in some EEG work, and we're also collecting some of our own data now to see if these model predictions actually hold, that we kind of know that this is how things should be, right? So in this particular task, we present two targets, two differently oriented gradings to the model. We have this first impulse just to see if we can decode those orientations. Now we show two other orientations. distractors. And then again, we have an impulse. Now, what I'll show you real briefly here is what we would expect at the second impulse. Because after the distractors, what we would hope that the model is doing, what the model is keeping in mind, is actually remembering the orientations of the targets, not of the distractors. But what we see in the STSP model, and again, this should not come as a huge surprise, what the STSP model is doing, it is actually at this time point during the second impulse, it is reflecting the orientations of the distractor and not of the target. Why? Because these dynamics, these STSP dynamics, they're slowly fading. So what they're going to store in that latent state is just the most recent thing that it saw, which in this case were the distractors and not the targets. Now, I alluded a little bit to this cueing mechanism that we have. So we're cueing to these frontal cortices, which is a little bit unfair, right? Because this signal never gets down to visual cortex, but we tried at least 3 different ways to cue. Amongst those are also cueing just directly onto early visual cortices. None of this produces kind of the behavior that we want or that we would expect from this STSP model. Now, of course, with the feedback model, it's a very different story. If we cue in the feedback model, we cue these frontal cortices, that information is able to propagate back to these early visual modules. And what we see there is that this feedback model actually can maintain just a target item. So what we're pinging out is information about the target and not the distractor in this case. All right, so that's it in terms of distractor resistance. We also looked at flexible selection. And so here is a bunch of EEG studies that have done a task very similar to this. So you get, again, 2 orientations. And then you are cued after a while to report one or the other. So in this case, you're cued to report the right stimulus. And so by the time impulse 2 rolls around, hopefully what you're holding in mind is only that orientation that was shown on the right, but not the one that was shown on the left. Now, if we look in the STSP model, what we see is that we can decode the cued as well as the uncued item. So that cueing, again, didn't really propagate. Also, if we use other cueing or other ways to cue the model, again, the same things happened, like we couldn't get the model to behave as it would need to complete this task. Whereas with the feedback model, we are able to, after the cue, we are able to then read out only the item that was cued, but not the item that wasn't cued. So the representation of the uncued item is now basically erased from the hemisphere that it was represented, both frontal and the visual. Okay, so what we see in this sort of theoretical model is that STSP, while it's of course a great model to get subthreshold information and to store it there latently in the network, and people have shown that, and it's also touted for that, it's a good model. I'm not trying to, discredit it in any way, but some of the flexible behaviors we ask our humans to do, and what we see from decoding in EEG experiments, it is somehow not able to do that well. So it cannot account for flexible selection, as in this last task, and it also doesn't appear very robust to destruction. And these are things, if you think about, or if you think it through, you would have predicted this because that's just how the mechanism works. But when you now try to relate it to the cognitive neuroscience literature, you can say that, okay, well, maybe then that's not such a good mechanism to use if you want to have some kind of short-term memory. That is flexible, right? It's also possible that both of these mechanisms are active at the same time. But what then the feedback mechanism and the neurofeedback model, what that enables us to do is that you have these durable memory signals that are sustained, but that are also reflecting now the task relevant content. So the target's not the distractors, the cued item, not the cued item. And so in general, we see that this mnemonic signals in early visual cortex that we can decode with fMRI, and we can decode through these pinging techniques in EEG, and also in monkey actually, that they likely reflect feedback and may not reflect this short-term potentiation of these network states. All right. And with that, then, I would like to thank you very much for your attention. And if you have any additional questions, I would love to ask them. This is my lab. I also want to highlight, by the way, Maria over here, who did the comparison circuit work, Noah over here, who did the modeling work at the very end. And unfortunately, I don't have a picture of Chaipat here. And I should also thank Albert Comte, who actually gave us like a touchstone when we were doing the ring attractor work, because we'd never done anything like that before. So that was super, super useful. And yeah, that's it.